In this video, we are going to be talking about the factors that affect an enzyme. Specifically, we're going to focus on temperature, pH, and the presence of inhibitors. When we talk about the activity of an enzyme, remember that it describes how fast this protein, called an enzyme, is going to be catalyzing a reaction, meaning turning the substrate, in other words, the reactant, all the way to products. There are three factors that we are going to be considering that affect enzyme activity. They include temperature, pH, and the presence of inhibitors in your reaction. When it comes to temperature and enzyme activity, understand that enzymes are going to have what is called the optimum temperature. And the optimum temperature is simply the temperature at which your enzyme is the most active. When it comes to the human body, understand that that temperature is 37 degrees Celsius because that's the temperature of the body. Understand that when temperatures go lower or higher, it affects the activity, but it affects it by different reasons. When temperatures are low, what you are affecting is the kinetic energy of the enzyme. So what this means is that at lower temperatures, the molecules, in this case the enzymes, are not going to be moving as fast. So since they are slowed down, they are not going to be able to catalyze as many reactions. Now, if we increase the temperature, specifically, even if we increase it all the way to 50 degrees Celsius, what happens is that now we are affecting the structure of the enzyme. So when we get to 50 degrees Celsius, the structure of the enzyme is affected by denaturation. And remember, when proteins denature, because they are unfolding, they are not going to have the ability to catalyze a reaction. Once you have unfolded a protein, remember that this effect cannot be reversed. So that's why it is important to understand that increasing the temperature and decrease the temperature are going to affect enzyme activity, but they're going to do it through different ways. When it comes to pH, we also have enzymes working at an optimal pH. But understand that that optimum pH is not going to be the pH of the blood. The pH of the blood is 7.4. But when it comes to enzyme activity, it depends on the location of the enzyme, what is going to be its optimum pH. When it comes to the definition of optimum pH, as you can see from the graph that we have, optimum pH is going to be the pH at which your enzyme has the maximum activity. Now, when we look at this, understand that enzymes, the side chains of some of the amino acids, are going to be affected by the changes in pH like we saw previously in the videos. Now, understand that when it comes to pH, at lower or higher pH, the enzymes are going to lose activity specifically because we are affecting things specifically, or the interactions at the tertiary structural level of enzymes. Now, to illustrate how different enzymes work under different pH conditions, we can see that in table 16.11, we have the optimum pH for selected enzymes. This table demonstrates that we are going to have enzymes that are going to work efficiently under acidic pH, and we can notice that in pepsin and sucrase, because if we look at their optimum pH, they are under acidic range. We are also going to have enzymes that are close to neutral pH, and that's going to be amylase and urease. And we even have enzymes that can work under basic pH. And examples of that are going to be trypsin, lipase, and arginase. And if we look at the selected list, understand that we also have different locations for them in the body. And when we look at the substrate that they interact with, they're even more diverse. Now we are going to proceed to talk about inhibitors. First, we are going to be talking about the different types of inhibitors, starting with competitive inhibitors. So, let's say that we have a particular reaction in which our enzyme is going to be interacting with its substrate, making the enzyme substrate complex. And remember that that enzyme substrate complex is going to turn into product, and we're going to regenerate the enzyme. So when we have a competitive inhibitor, as you can see, the inhibitor is illustrated in the bottom of the figure. The inhibitor, in a case of a competitive inhibition, is going to have a similar shape in terms of molecular structure. So shape, because remember that these inhibitors are actual small molecules. So shape in quotation marks, but in reality is going to be molecular structure to the substrate for the enzyme. And as you can see, because the structure of the inhibitor, it is very similar to the normal substrate of the enzyme, a competitive inhibitor is going to sit in the active site and prevent the binding of the original substrate. When that happens, then this is going to inhibit the enzyme catalyzed reaction. Now, there is a second type of inhibition, and this is called non-competitive inhibition. So in a non-competitive inhibitor, as you can see, this is going to be the normal reaction on top. Okay, which we have an enzyme reacting with the substrate, making the enzyme substrate complex, regenerating that enzyme and forming product. Now, in the case of a non-competitive inhibitor, notice that the inhibitor does not have the same, and I'm going to put in quotation, shape, but in other words, molecular structure when compared to the substrate for the enzyme. So what happens is that whenever we have a non-competitive inhibitor, it's going to bind, okay, at a different site. So somewhere else in the enzyme that it is not the active site. And overall, having this inhibitor bind a different site than the active site is going to create the enzyme to have a different shape. So now the substrate cannot bind the active site because it changed. This type of binding that it is different from the active site is called an allosteric site because it's different from the active site. And then, because now the substrate cannot bind, okay, the active site of the enzyme, that's how the enzymatic reaction is inhibited. We have one more type of inhibitor, and that is called an irreversible inhibitor. When it comes to irreversible inhibitors, remember that this molecule is going to cause the enzyme to lose all of its activity, okay? Often these irreversible inhibitors are going to be toxic substances that are going to, in a way, destroy the enzyme, but it's not that it's going to make it crash. What it means with destroy is that it's going to lose its activity. Now, the reason why they're called irreversible inhibitors is because of the formation of a covalent bond with an amino acid side chain, and that's what is going to be preventing a catalytic activity. The formation of the covalent bond itself can be at an allosteric site or it could be in the active site. So to give you guys some examples of irreversible inhibitors, some of them include nerve gas, insecticides, and even antibiotics. So when penicillin was discovered, it actually revolutionized the field of medicine because this gave the opportunity to have bacterial infections to be cured. Now, since the creation of penicillin and the discovery of the mechanism of how it does that, as you can see, since bacteria have evolved to be resistant to these medications, other versions of penicillin have been created. As you can see, some of them include ampicillin and amoxicillin. And if you have some type of infection, at times you will be receiving a prescription on those medications. Now, to summarize overall, the difference between competitive inhibitor, non-competitive inhibitor, and irreversible inhibitors, all of that is summarized in table 16.12. As you can see, for this, you must know the information in this table for the final exam. Here, what we have is that the competitive inhibitors, non-competitive inhibitors, and irreversible inhibitors are described in terms of the shape of the inhibitor, the binding to the enzyme, and if they're reversible. The only thing that I haven't mentioned till now is the reversibility for each one of the inhibitors. Of course, when it comes to the last one, the irreversible inhibitor, that one is permanent and is not reversible. But it is interesting to know that competitive inhibitors can actually be reversed simply by adding more substrate. In the case of non-competitive inhibitors, they are not reversed by adding more substrate, but you need to add a chemical agent that is going to physically remove that inhibitor. And I just mentioned that irreversible inhibitors, again, those changes are permanent. We're making a covalent bond, so they cannot be reversed.